Hello, and thank you for joining IDEX Health and Science for our series on optimizing fluidics for IVD. Today's segment covers valves that are commonly used in IVD. We have a number of product experts with us from IDEX, and in addition, we have a guest who's a design engineer and system developer. And our format for this program will be a brief overview, an introduction of the participants, followed by a roundtable discussion. That will last approximately 20 minutes, after which we'll answer questions from those of you listening on the phone. You can begin keying in your questions anytime into the box provided. If you're viewing this program as an on-demand webinar, you will hear the question from the original webcast, and later in the program, we'll give you an email address to send us any remaining questions you may still have. I'll take a moment now to go around the room and introduce each participant in today's webinar. You'll also see on screen some additional details about each person's fluidic experience and product specialties. Our guest today is John Mignolo. John's a mechanical engineer and has been a system developer for several instrument platforms with more than 25 years of experience. Currently, he's the principal consultant at Fluidics Design Incorporated. Corey Phipps is our valves product manager. Corey has a scientific background as well as deep experience with our degassing and debubbling product lines. Frank Melgano is Director of New Product Development at our Bristol, Connecticut manufacturing facility. Frank has a strong background in R&D and especially the engineering required to precision machine plastics, such as our Eastern Plastics manifolds. These are now widely used in IVD. Frank also has a lot of experience integrating valves into manifolds. Darren Lewis is a scientist by training, and Darren manages research and technology development for IDEX Health and Science, and has spearheaded quite a few of the integrated systems we have recently been releasing. Mechanical engineer Jeff Stokes has extensive experience designing fluid handling hardware from atmospheric liquid handling robots to ultra-high pressure LC systems, pumps, and valves. Here at IDEX, Jeff leads our system integration projects. I'm Bob McCarthy, R&D Director at IDEX Health and Science. Throughout my career, I've worked in a range of instrumentation from cell culture systems to drug discovery systems in regulated environments. All 5 segments of our series of webinars, Optimizing Fluidics for IVD, will be available for immediate playback on our website. Just visit webinar.idex-hs.com, where they're also available for download as podcasts. One quick note before we get started with our roundtable discussion. We want these discussions to be open and informative for the engineers who are our customers. That said, because we deal with innovative, often patent-pending technologies, in most instances we're bound by non-disclosure agreements. Now, that means we won't be able to give you the names of recognizable partners or platforms. We'll refer to them generically as, say, a leading hematology manufacturer or a life science assay instrument. The stories are real. The names have been changed to protect everyone. Valves have always been a major consideration in the fluidic circuit, but with the advance of micro and nanofluidic technology on scientific instruments, valve usage has become more ubiquitous and more inventive in some cases. In IVD, we have seen a growing number of customers asking us to partner with them to provide more design and integration support to help identify more optimal solutions in these more demanding applications. Sometimes the requests are driven by time-to-market needs, or perhaps a lack of available internal resources. Increasingly, we see the need to create subsystems to reduce the number of vendors that our customers require. But often it's simply a desire to brainstorm with people who optimize fluidics every day. On screen, you'll see we provided quite a number of different solutions. Discrete passive valves like check valves in the upper left, or actively controlled valves like solenoid valves in the center. Also becoming popular are rotary shear valves that minimize fluid pumping during actuation, among other benefits. So which fits the application need? Choosing the right valve is one part of the equation to optimize a fluid flow path. So what does it depend on? Well, we can discuss tailoring solutions for precise handling and flow redirection for samples and reagents. That may require a very different type of valve than, say, bulk delivery of fluid or removal of waste fluids after a reaction has taken place. So then there is the medium that is being moved. Is it clean, neutral pH, or more challenging fluid for the valving mechanism? Types of control also depend on the application requirements. So Jeff, can you give us a few application examples to get us started in this discussion of how we begin thinking of the right valve and trade-offs to consider to optimize the system for our customers?
Sure. There's really a lot to consider when you're talking about IVD, the hardware, and the liquids. You know, for reagents and buffers, for example, you really need to consider the pH and the chemistry of this fluid and select components that can handle the everyday exposure. But the components may also need to handle even more aggressive occasional exposure, like during a preventative maintenance, you know, like some concentrated bleach to clear out any residuals that are in there. If you look at the actual fluid handling itself, I usually try to break the applications down into two categories. There's precision handling and then there's bulk handling, like you mentioned. Precision handling, for example, would be handling samples, moving them through the fluidic path, or the precise handling of reagents for addition. These operations usually deal with very small volumes, a few microliters up to a few hundred microliters, even more. And they need to be very accurate and repeatable to ensure that the assay results are reliable. So CVs for these fluidic operations, you know, should be and could be required to be less than 1%. So the selection and placement of these valves in a sample or reagent system is critical to ensure that there is no carryover. Bulk fluid can actually also still fall under the precision handling category. For example, when reagents or diluents are mixed or created on board the analyzer from concentrates and a water supply, these reagents and diluents are directly involved with the analysis and have the same tight CV requirements as the sampling handling. So, you know, valves for precision handling systems need to be carefully selected to meet the required performance. The other larger category, typical bulk fluid handling operations in IVD, are really not as stringent. For example, wash systems or waste systems. For wash systems like probe wash stations, flow rates and total volumes need to be controlled, but typically do not have accuracy or precision requirements. Waste systems are at the other end of the process and typically have very minimal requirements. If we start to look at, you know, liquid handling considerations, when it comes to handling the samples, as we mentioned, we need to consider the properties of the sample. You know, what components or materials of, you know, will the sample need to touch? Will there be any interaction? Will the viscosity of the sample be a factor? Some of these samples are very viscous. Will it crystallize if it's not flushed out after use? Will a bleach cleaning be required, like I mentioned, either daily, weekly, or monthly? Are there particulates in the sample? The components that will see these fluids need to be very carefully selected. Frank, can you talk about what you've seen for buildup in these valves?
Oh, sure. Thank you, Jeff. So here at the picture that we're looking at, the black component in this picture is a rubber diaphragm seal in a typical two-way or three-way chemically inert solenoid valve. The diaphragm is the moving part that's actuated by a solenoid to allow shut-off or fluid flow through the valve or orifice, just like turning on or off the water in your kitchen sink. In the middle, you can see a small circular impression in the center of the picture where the rubber diaphragm contacts the seat. And next to that, the arrows indicate salt buildup on the rubber diaphragm. That commonly results when salt-based solutions and buffers are allowed to empty out of the valve cavity and allowed to dry. And when that occurs, the buildup can cause a flow restriction past the diaphragm seat. It can cause the diaphragm to stick to the seat so the valve may not energize or operate when called upon. And also the valve diaphragm may not seal the seat off completely, which can permit fluid leakage in the de-energized position. So salt buildup can be prevented by keeping the cavity fully wetted or rinsing an evacuated cavity with demineralized or deionized water after use. Okay, next slide. And then this is a picture of a plastic solenoid valve seat. In the center, there's a 1.6 millimeter diameter orifice. And looking at the arrow, you can see that it's partially blocked by a salt crystal. So here, the formations are likely to cause a diagnostic instrument not to be able to perform properly, and this builds up slowly over time and overuse. So once again, if the instrument's not going to be used for an extended period of time, maybe at the end of the day or over a weekend, there should be something in the designer's protocol to make sure that the system is flushed out. Okay.
So applications aren't the same, and all instruments aren't created equal. They have different challenges. One solution, I guess, to simplify the system when possible is with passive valves actuated by the fluids themselves rather than a motor. Darren, you've had experience specifying check valves for systems in IVD. When are check valves the right choice?
So check valves are often used when you want to achieve unidirectional flow. So that is flow in one direction and have it prevent flow from going in the reverse direction. You also use them when you need a fairly small footprint and you don't want to have any electronics required to actually drive the valve. Typically, check valves work on a mechanism that involves a ball in a seat or some kind of flap that will actually obscure an orifice to prevent flow in the reverse direction. So with that said, they require actually a relatively clean fluid stream with fairly low particulate generation or other material that can actually come out of solution, because that can actually impact the ability of the valve to check and to prevent leaks in the reverse direction. But they're very handy. They're used in analytical instrumentation and in the diagnostics area for places where you want to automatically control flow in one direction. So that's pretty much where you'd actually find them used, Bob.
Yeah, great. So design trade-offs sometimes include the need, though, to force the path closed or open it, right, and do that more directly. And solenoid valves have seen a lot of application here. John, I know there are many types out there, and some of them are displayed for our listeners to see. But could you walk us through a couple of examples of how and why they are applied in IVD fluidic circuits?
Yeah, there are a few different types of solenoid valves out there, and you probably can find a solenoid valve that will fit many of your applications. One of the most basic valve is a standard poppet valve that has a little elastomer seal that rides on a poppet up and down every time the valve is energized. These valves are good for when you just need a clean fluid wash. The downside about that valve is everything in the internal part of the valve is wetted, so you got to really watch your materials, compatibilities, and also if there's anything that might gunk up the works of the mechanism of the valve. Then you have this type of similar poppet valve, but that uses an isolation diaphragm, which at least separates the valve operator from the fluid. And that could do very well with wash applications, dispensing of clean fluids. You still have to worry about particle-filled solutions because they may form around the seat and cause a leak. You also have what's called a standard pinch valve, which is just a tubing that gets pinch closed. And one of the great things about a pinch valve is there's no internal volume and there's nothing internal getting wetted besides the tubing itself, but you have the maintenance of the tubing. And another standard valve that's out there now is a rocking diaphragm isolation valve, which is one of the better valves when you have particles because it has a great sweeping flow through the internal part of the valve. Once again, the diaphragm separates the main flow area from the operation of the valve mechanism. And the way it's designed, you get a good sweeping flow, very little dead volume inside that valve. Typical standard valve there for the industry for years is a Teflon isolation diaphragm valve, which still has its applications in certain solvent needed flow conditions. And one of the last ones that doesn't get a lot of play out there is what's a lever valve, which is an elastomeric covered beam or lever that snaps between ports. This valve is also very good in waste solution handling systems and handles particles very well. Frank, can you elaborate maybe on an application of these valves?
Sure. In many of the IVD applications that we work on, hematology and chemistry analyzers, solenoid valves are a very popular choice. Most of the valves that we populate our manifolds with are of the isolation style, so that there's inertness between the diaphragm and wetted materials and the customer system. So for on-off control and directional control, the typical 2- and 3-way solenoid valves offer a small package size, so there's high density, fast acting, long life, and low price for the value.
Great. So very soon we're going to talk about an overview of an entire system. But before we do, Corey, some challenges are related to dead volume or maybe smaller flow passages or even small dispenses where the valve actuation itself may be a larger factor in precise dispensing. So can you walk us through a short outline on rotary shear valves, briefly the difference in how they operate and their potential benefits?
Sure. So rotary shear valve operates by having two flat faces, one of which that has holes that are connected to the various fluids that will be handled by the valve. The other has grooves. So as the valve is actuated, those grooves slide over those holes, connecting the various holes and therefore changing the flow. So as a result, as was commented earlier, there's no pumping. There's nothing pushing or pulling on the fluid as it actuates. So it can be an advantage in very precise dispensing. Also then results by design in low internal volume and very low dispersion or carryover by the design of the flow passages. Another thing that you can include in a rotary shear valve as opposed to the check valves and ceramics is check valves or solenoids is the fact that you can use ceramic surfaces, so you get a harder surface that is a lot more tolerant to particulates. So in applications where particulates are an issue, often rotary shear valves can be a good solution there.
So maybe we could go to the next slide we have. We thought it would be really interesting to talk specifically about an integration of a number of different components. So, Jeff, would you lead us through this particular one? There's a couple here we want to go through and talk about where certain valves may be more beneficial than others, but there are trade-offs to all, right?
Sure. So the next two slides are actually going to compare two systems and some different hardware that was chosen, specifically valves. So this first diagram is an example where solenoid valves would be a good choice for this application. In this case, there's a pump shown to the left. It's a gear pump. This pump is capable of continuous and high flow rates, much more than the next example is going to show. In this case, the pump feeds a manifold which has multiple solenoid valves mounted direct to the manifold. This could be a wash system for a large analyzer, for example. So the big advantage of solenoids here is that any valve can be energized at any time. So this means that one valve could be powered, or any of the valves or all of the valves could be powered, and they could be constantly changing, is what's typical in a complex analyzer. So the particular state shown here shows one sample wash station, two probe wash stations, and one dispense pump and probe being flushed by the system. So this particular system actually monitors solenoid valve activity and fluid pressure and adjusts the gear pump accordingly. So in a complex system like a large analyzer, solenoid valves can provide a simple solution to provide the flexibility that's needed. The next example is for a shear valve application. This example is going to show how a shear valve would be a better choice than solenoid valves. So on the left is a precision dispense pump. In this case, this pump is plumbed directly to the common port of a selector-style shear valve. This pump valve arrangement allows the discrete selection and dispensing of any of the reagents shown above the valve. So the pump would aspirate the particular reagent in. After aspirating, the valve would rotate to a new port and allow discrete dispensing. So there's benefits of this configuration with a shear valve. One is you have a fully swept fluid path with zero carryover of the fluid. You can configure this with either high pressure or low pressure capability. It's very low volumes in the valve, and there are some very good material choices available.
All right. So any other comments from those on those two flow charts for folks? Any additional information? And if not, let's talk about what's next for valves and IVD. Where do we think this field is headed? More or less control for valves, in other words, the check valve versus solenoid or rotary valves actuated by motors, higher throughput driving faster response or actuation. Corey, you've seen some of the different requirements IVD customers have challenged us with. Where do you see the market trending?
I'd say the first comes in mind is getting smaller, both smaller in overall package size, trying to reduce the size of the instruments, but also smaller in terms of reagent use and basic general solutions that go through the system. So all components, essentially the internal passages getting smaller to support that lower usage without adding additional issues to the instrument. I think that would be the primary one that I've seen.
Mm-hmm. Darren, in your experience, or in your, where you see your engaging customers, where do you figure IVD is headed at this point in time?
Well, Bob, what I'm seeing fairly often now is that customers are actually engaging us at the design level from the outset of the design of the instrument. Used to be, I think, customers would do a lot more upfront work before coming to us, but I think with sample volumes getting smaller and precision requirements increasing, people are tending to actually come to us for ideas earlier on in projects, I think. Overall, I think I see that as a bit of a trend in the industry.
Agreed. John, anything to add for where you see the IVD market space heading?
Same as Corey and Darren. I definitely see, looking for smaller packages, valves that are smaller but also could give the flow of a larger valve. And also in most of the IVD industry, they definitely want to use less reagent, less of the liquids on the system to save overall cost.
Mm-hmm. Yeah, I guess without leaving you up, Frank, do you have anything to add on trends for the marketplace?
Well, I just think that there are a lot more customers out there looking to integrate more components into their fluid control system. So the valves, the pumps, degassers, debubblers, and also flow sensing and feedback. Am I getting the right amount of flow that I expected? Is it flowing at all? Are some of the the questions or concerns that we see some of our designers challenging us with.
Finally, Jeff, from your point of view?
Yeah, I think I'd echo a couple of people there. John, with the smaller sample size, trying to get more analysis out of a smaller sample, and Frank and Darren, that we are seeing a lot of OEMs that want to consolidate a lot of their components into subassemblies, into manifolds to minimize fluid paths and try to minimize the amount of volume that's consumed inside the instrument.
Mm-hmm. Okay, so we want to leave time for your questions, so we'll conclude the roundtable portion of our webinar. For information on how IDEX Health and Science can optimize fluidics for your instrument, check out www.idex-hs.com. While you're there, you can request our new instrument design resource. It's free and it was developed specifically for design engineers like you. So let's go to our questions, and thanks to those of you who have sent in your questions for our fluidic experts. First up, this question came in related to selection of valves. So is there a decision tree that I should consider when selecting a valve type for IVD applications? Jeff, you want to field that one?
Sure. What I would do is consider some of the things we've discussed today. Is it's not really a tree, but it helps you go through and decide what features in a valve you're looking for. What's the chemistry and or sample that's going to be in this valve and exposed to the materials? What kind of pressure is this valve going to see? Is it going to be atmospheric sample handling, or could it see higher pressure like in a wash system? Is carryover a concern? Some valves are better for carryover than others. Same thing for dead volume. You just have to go through and start answering these questions. How will this valve interact with the rest of the system? Is the actuation time of this valve important? What controls are available, or what controls do you want to develop for your system? What power is available? What is reliability a concern? And usually most importantly is what's the cost of the valve?
Mm-hmm. So here's the next one. How can I diagnose a problem with a valve? Darren, can you give us a little bit of insight on that from your experience?
Sure. Most of the time when I'm asked to look at a valve problem, I go through a few separate steps. First thing I'd probably try to do is isolate the valve from the fluid path so you can actually see how the fluid's moving through the valve in absence of things around it. And what you want to really look for are the failure mechanism of the valve. For instance, Is fluid flow going into the valve and then leaking out internally in the valve, or leaking out through a path that you weren't expecting, or is there blockage in the valve that's actually preventing fluid flow from happening? The second thing I would do after sort of evaluating where the fluid's going is to look and see how the valve actuates. So is it either rotating properly for a shear valve, or is it clicking open and close properly for a solenoid type of valve? And you probably want to need to be operating the electronic circuit that runs the valve in order to do that. Once I took a look at all those things, I'd actually contact the vendor, if that was us or some other manufacturer to help them work with us on the problem, take a look at the fluids that have been passed through it, and maybe do a brief review of the history of that valve to try and understand what could have gone wrong with it. And that's pretty much the structure that I'd follow to evaluate a problem valve.
Okay, good. Here's another one. If cross-contamination is of paramount importance, would you recommend rotary valves? Corey, I'm going to let you take a look at this and see where do you think rotary valves fit in this, and it does have benefits over other choices.
Yeah, certainly if cross-contamination is a primary concern, a rotary valve would be a good choice. It'll depend some also on a couple other factors. If you can obviously, if you have the opportunity to flush, if the timing is not critical, maybe then you may consider using other valves as well. Another potential factor there would be to the point that Jeff pointed out. We were showing the schematics earlier, the various diagrams of the systems. Is there a requirement for random access to what it's doing? If there is, then a rotary valve, you may have to make a compromise between the really good performance of a rotary valve with respect to carryover and cross-contamination and the need to have those random access of the positions in the system. But really, it's one of those things where typically we would work with a customer and understand their complete application, how they're applying it, and then you make a recommendation. But in general, I would answer that as a yes. Rotary valves would be a good solution for that.
One quick follow-on for you, Corey, that's just come in. How does biocompatibility relate to shear valve design?
Biocompatibility is a little dependent on who's asking. But overall, I'd say in general, it's definitely, I think everyone would agree that it's elimination of any iron from the flow path that might interact with a biological molecule. And then for the most part, the way we approach it, we also would eliminate any metal at all from the flow path. So typically then going with inert polymers, such say as a PEEK or Teflon, or going to a ceramic type approach. Excellent.
We're running a little short on time, so I want to make sure that you all know that any questions sent in, we will answer them. Please continue to forward them in. To review all of the archived segments in this webinar series, visit webinar.idex-hs.com. For additional questions, you can contact our guest, John Mignolo, at john@fluidicsdesign.com. To reach one of the participants from IDEX Health and Science, call 866-339-4653 or email customerservice.hs@idexcorp.com. It's been a real pleasure going through all of these discussions with all of you. We would certainly like to join with you in the future on some of the future webinars. So for IDEX Health and Science, I'm Bob McCarthy. Thank you very much for joining us.